thank you for inviting me. And as we said before, there will be diversity is important. So as a woman before, a man now. Animal, plant organs, organs in animals and organs now in plants. And okay, it's working perfect. Okay, sorry, just need to look at the pointer. Okay, perfect. And so I think that it's interesting, even if most of you are probably animal biologists, to think about plants as well. Because obviously plants have evolved a mechanism of multicellularity independently from animals. So that is going to be influencing a lot the way they grow. But at the same time, they are submitted to the same physical laws that animals are submitted to. And I hope you will see that the concept that we have in the animal field can be also translated, in the plant field, can be translated in the animal field, and vice versa, obviously. And so I'm very happy to show you the work we are doing in our lab to try to understand basically how plant organs grow, using the seed of the plant model Arabidopsis as a model system. So the question we're asking is about morphogenesis. So that's about how plants or animals can acquire their shape. And for many years, because of genetics, people were looking at that left part here, which is how from a group of cells, with the action of patterning mechanisms, you can get patterns of cell identities. And then they will be translated into shape changes. But then the more we advance in the field, the more we realize that it's not so easy, actually, to pass from a patterns of cells to a given shape. And for that, you need to look at growth, which is kind of a complex process, a biophysical process. And also, here I've shown that linearly, but actually, things are not linear. We saw it in the talk this morning before the break, that basically growth and patterning occur at the same time. And so you have ways, cells have evolved way, to actually sense changing in shape so that you can provide robustness to morphogenesis. And one of these ways to sense shape changes is through mechanical signals, and I will show you later. So if we go in the plants, for me at least, the biggest difference there is between a plant cell and an animal cell that's going to be very important to understand how a plant cell is growing is the presence of a cell wall. So you have a cell that is encased in a very stiff cell wall. If you want to grow, you have to change that cell wall. And so that cell wall restricts the growth, and the growth of a cell is promoted by turgor pressure. And turgor pressure comes from the accumulation of small osmolyte within the cell. And because you have that cell wall that is extremely rigid compared to animal cells, you can have very high level of pressure, like up to, I think, 10 times what you have in your air chamber in your car, in the wheel of your car. So plant cells are really pressurized compared to animal cells, and that is due to the presence of a cell wall and its structure. So the cell wall is a very complex structure which is made of various types of polysaccharides, and you have, for instance, here cellulose fiber, and the tensile strength of cellulose fibers is the same order of magnitude as steel fibers, like of fibers of steel that would be the same size. So it's really, really stiff, and those fibers of cellulose, they're embedded in a matrix that is made of all other type of polysaccharides, such as pectins and hemicellulose. And so if you want to change growth, you need to either increase turgor pressure, or most of the case, you need to change the mechanical properties of your cell walls. So that's for the cellular scale, but also the presence of a cell wall also involves other things compared to animal cells, is that the cells are glued together with their cell walls. They can't move, so there is no movement when you talk about growth. They can't intercalate, and in plants there is rarely cell death. There is only occurrence sometimes, but it's very rare. So because, and all of that, at least the two left part are due to the presence of the cell wall, so the cells are glued together. And that means that when you take a tissue, for instance, or even an organ, the way one cell is going to grow is going to influence the cells that are adjacent, because they share the same walls, and if one cell is trying to grow, that's going to pull the other away. And you can see that through those easy models. For instance, here you have a tissue, basically. The cells are not really represented, it's the little round thing there. And let's say all the cells are trying to grow towards the center. What will happen actually, because they need to keep being next to each other, is that from a 2D flat structure, you will be making a 3D shape. You can also do the same and looking at the mechanical interactions between different tissues. So here I have two layers of cell. The one on the top is trying to grow more than the one at the bottom. But the two are stuck together, so they need to maintain their integrity. And the only way they can grow this way is basically to fold like that. If you fold like that, the surface is going to be higher in the top layer than in the bottom layer. Okay? So you can see how you can make shape, different shape through mechanical interactions, or in this paper they call it actually what we call resolution of mechanical conflict. You have different types of mechanical properties between cells that are going to give them different types of growth, but because you keep the integrity of the tissue, and because of the cell wall, you are going to make those complex shapes. And so mechanistically, for many plant organs, the way the plant organs are growing is very easy to understand. It's basically they are balloons. You have inner tissues that are compressed, and through turgor pressure are trying to push, basically, to promote the growth. And then growth is restricted by the mechanical properties, especially the cell wall properties, of an outer layer, which is often the epidermis. And you can very easily visualize that through those canonical, like very historical experiments in sunflower stem. So those are hypocotyl, it's called its embryonic stem. See, you take the stem, if you peel off the outer tegument, so the outside, from the inside, what you will see is that the outer tegument, because it was in the tension trying to restrict growth, when it's separated from the inner tegument, is going to shrink, whereas the inner teguments that were compressed, they are going to expand. And the properties, so at the end, people don't focus too much on this role of those inner force. Also, I will show you in the seed for us, that's very important. But they will focus on how the epidermal walls are going to restrict the growth. And what you can see is that in many organs, the outer epidermal wall, so that one here, if you follow my screen, is thickened in order to resist to the pressure of internal tissues. And if you change those properties, you're going to change locally the growth, and that's going to make bumps, for instance, in your balloon that is growing, like that organ here. But then there is another interesting thing in that model, is that you see here those arrows, they point at kind of forces, that as you see, depending on the position of the cell within the organ, that cell is going to be submitted to different patterns of mechanical forces. It's going to be either pulled or compressed, and that is depending on position, but also on the overall shape of the structure. If you change the shape, you change the pattern of forces. And we know that in plants, like it's the case in animals, cells are able to sense forces, to be able to sense if they are stretched, if they are compressed, and that affects their behavior. So that can affect the organization of the cytoskeleton, notably the cortical microtubules. That can influence the localization of specific proteins at the membrane, or can even induce the expression of specific genes. You can see there actually we were compressing the organ, and you can see the localization of microtubule, or here compressing the organ, you can see the induction of specific genes. And from that you can think that basically forces are a way, mechanical signaling, are a way for cells to assess on shape changes that occur at the organ scale. Because if you change the shape of your organ, you change that pattern of forces, and the cells can sense it. And so our question then is how the mechanical properties of the cells, so turgor pressure, cell wall composition, and then mechanical properties, but also how the response of the cells to mechanical forces contribute to plant organ growth. And to study this question, we are using in our team the seed of Arabidopsis as a model system. Why? It's because first, it's a very simple system. It's kind of a flat ovoid, a flattened egg, if you want. So it's much easier, you will see model after, it's much easier to work with that than with complex organ, if you want to be really mechanistic. But also, there is a very interesting feature of the seed, is that the seed is composed of 3 genetically and physically separated compartments. So the seed has an embryo, which is here very tiny red blob there, that's going to grow, fill up the space inside, and that's going to give rise to the next generation. And that embryo is a product of fertilization that is diploid. But then in plants, you have what we call double fertilization. So the pollen tube arrives with 2 nuclei that are going to fertilize 2 cells, and there is a second fertilization is going to give rise to the endosperm, which is all the yellow thing you see inside there. That is also then a zygotic tissue, product of fertilization, and that tissue is actually triploid. And surrounding both embryo and endosperm, you have the seed coat, or testa, which is a set of maternal tissues that are there to protect the seed, okay, the ovule at the beginning and then the seed. And those ones are maternal tissues, so they are not product of fecundation, and they are diploid. And as you can see on the skin, so the seed is growing quite fast, within like 6-7 days, fast for plants, sorry, I should say actually, compared to Drosophila. But within 6 to 7 days, you have a seed that is pretty much the size and the shape that it will have of mature seeds. And you can see within those first days, the embryo is very tiny. It doesn't really play much role in defining that seed size and shape. What matters more is that interaction between the endosperm and the seed coat. And then that define that inner cavity here that is going to be filled up by the embryo as it's going to degrade the endosperm at later stages. And so at our institute, before I actually joined, there's a team of Arezki Boudaoud and the team of Gwyneth Ingram that I'm part of. They were trying to look mechanistically, from a mechanical point of view, as how is that really working. And to do that, what they did first is to use a nano-indentor. So it's kind of, the technology is different, but the output in a sense is the same as an IFM, but for larger indentation, where you press on a seed and you measure the force that you have in return to see is it stiff, is it soft. And what they could see is that the seeds are pretty stiff, but at early stages, when they are still growing, if you plasmolize them, if you remove the water from inside, by putting them in a medium of high osmolarity, you will get very soft seeds. So that shows you that basically the seeds are a balloon that is inflated with water. And that balloon is actually, the water is actually in the endosperm. Because if you then pierce the seed up to the endosperm to remove the liquid that is inside, you see that almost all of the rigidity is also gone. And then Arezki Boudaoud also developed just like kind of a model with an equation from which you could pass from rigidity measurement with the indentor, and assuming that the seed is like a shell, an inflated shell, and taking into account the curvature of the seed, you can get ideas of what's the pressure inside the endosperm, inside that seed. And what we could see is that it's high at the beginning of development, and it slowly decreases over time. So basically, we have a balloon that is inflating with liquid going inside the endosperm. That's giving up the pressure. Then to restrict the growth of the seed, you expect that the shell, which is, in our case, the seed coat, so those maternal tissues that are at the exterior, will restrict the growth. And so that seed coat is composed of 5 layers of cell that we define as the outer integument and the inner integument. Okay? Outer are the more like outside, basically. And they are separated by a wall which is called wall 3. And what was fun in the team of Gwyneth Ingram is that basically that wall over time is thickening, okay? Which we see thickness can be related sometimes to restriction of growth, at least in other organs. And also, the composition of this wall is changing over time. It's accumulating dimethyl esterified pectins, which are usually a mark of stiffening of the cell wall. So we could think that basically, really the seed is a balloon. If you have one thing to remember from this talk, it would be that that organ is a balloon, where you have pressure inside that is promoting growth, and then the shell, the layer that is surrounding those seeds is stiffening in order to compensate for the pressure. Or it's like a single cell, but on an organ scale. So how can you then coordinate those two? And we think here that there is a role for mechanical forces. Because if you think about your balloon, as you are inflating your balloon, you are going to pull on the cell at the, on the seed coat. You can see this effect because if you do again the seed pre-kick, so that's a section of a seed, you see the cells in the first layer there. If you pierce the seed at quite far away actually from there to remove the liquid that is inside there, you see the cells are going to shrink to be more, actually increase in thickness, because it's a bit like what I showed you before in the hypocotyle. They are pulled by, so they are under tension by the pressure of endosperm expansion. And what's nice with these cells is that we know they can sense that tension and respond to it, at least from a transcriptomic level, because we've identified a gene, and there are probably much more genes that are influenced, that is specifically expressed in the second layer of the seed. And that's actually the layer that's producing a wall that is stiffening and thickening over time. So that gene is involved in the degradation of a growth hormone called gibberellin in plants. And that gene is a negative regulator of growth, because if you don't regulate its expression, here you have a line where you have artificial microRNA to inhibit its expression, you get larger seeds. And what's nice also is that if you compress the seeds within the fruit, so that's a little fruit, that's like a bean if you want in Arabidopsis, you have seeds inside, you compress the fruit, you can induce the expression of the gene. And so based on that kind of data, we can propose a model, a very simple model that we referred as an incurrent mechanosensitive feed-forward loop, where you have a balloon that is inflated with endosperm pressure that's going to promote the growth, but at the same time the tension it generates in the seed coat is going to be perceived by the second layer, so those light blue cells, and they are going to stiffen the cell wall in return. And that is going to inhibit growth. Okay? And you see here the ID, we use the ID from the gene regulatory network of using that incurrent feed-forward loop in gene regulatory network. Is A is inducing C, but at the same time is inducing B that is inhibiting C. And so we use to see, we try to see if that concept actually could be applied to mechanical processes. And what we wanted to see is, can we use that model here to explain the growth pattern of the seed? So Arabidopsis is producing tons of seeds. Within one fruit, you have like around 30, 40 seeds. And you have Arabidopsis, one plant is producing like many, many fruits, so that at the end you can have up to 10,000 seeds from one single seed. And so you can sample seeds every day, marking when is the flower fertilized. And then you can have those patterns of growth, which is showing kind of a typical S-curve, very similar to what you have in bacteria, actually. And if you derive that to have a growth, you see that growth is peaking around 2 to 3 days, or 1 to 2 days, and then it's slowly decreasing until it reaches a plateau around 7 days. And so in order to see if that ID, mechanical model, could work to explain that growth pattern, we teamed up with a physicist, with a permanent researcher in the modeling team at the RDP, in the Mosaic team, who is called Olivier Ali. And basically, we tried, again, like we saw in the talks before, to do the model as simple as we could of a seed, where we say the seed is just an inflated shell. Okay, there's pressure inside. It's going to lead to some stress in the seed coat. Okay, that is going to lead to some strain, so deformation of the seed coat to increase the radius of the seed. But at the same time, we implemented that function here that says that stress is going to stiffen our cell wall, and that is going to prevent them from being deformed, basically. And we use very simple equations. So we use the Lockhart equation for that part, which is a kind of usual equation that people use for plant growth, basically. And then we said we don't really know how the stiffening occurs in response to stress, so we just model that as a Hill function to say it's kind of an enzymatic reaction that is stiffening the wall in response to stress. And because our system is very simple, what we could do is to make simulations, changing all the parameters of the equations. We tried 500,000 parameters, and then we selected which simulation give us the same kind of pattern of growth that we see in the experimental data. And then we looked what type of parameters do those simulations have so that they can fit the experimental data. And what we see is that basically we need some specific features for the stiffening function in response to stress. It needs to be quite strong, okay, to occur rather fast once it's triggered. But an interesting thing is that you have a threshold activation. So in our system, stress is a function of radius. It's a physical principle. The bigger the CD is, the more stress you have. And stress is increasing in our system. And so you need to have a threshold of stress for the stiffening to occur. So another way to see that thing here is to look at the stiffening function in response, the stiffness of the shell in response to time in the simulation. You see at the beginning it's not stiffening, and after a few days it's increased and you reach a plateau. And that's what's inhibiting your growth in the system. So then we tried to see if we could correlate that kind of pattern, see that kind of pattern with in biology. And as I showed you before, one thing that we see is occurring in our seed is accumulation of dimethyl esterified pectins that we can see with antibodies. So here is an antibody. So Vincent Bail, who is working in another group in the RDP, he developed a plugin in Images so that we could segment the cells in the seed coat on our sections, and then we measure in each wall the signal that you have in the antibodies. And what we can see is that if we do look at dimethyl esterification of pectins with two types of antibodies that are a bit different, that one is partially dimethyl esterified pectins, and that one is completely unesterified pectins, we can see a kind of pattern of accumulation, a bit like here, where at the beginning it's quite low, and then there is that S-slope until you reach a plateau. That one, being an esterified pectin, is a bit later. You can see you don't reach a plateau before in this time. But you see the pattern look kind of similar. But that does not tell you that our model is right. When you do model like that, if people do a model with simulation, you know, a model is like, it can reproduce, the data is good. If you want to test your model, the nice thing to do is to bring a new set of data, completely independent from the model, and see if the model can behave the same and reproduce that new set of data. And what's nice with the seed is that seed size is quite important for agriculture. So people have done tons of screens, genetic screen, and they have isolated mutants of seed size. And so we characterize one of those mutants, which is called IQ2. So it encodes for a receptor-like kinase, we don't know the ligand actually, that is part of a pathway that's expressed specifically in the endosperm. So it's specific to endosperm, it's controlling endosperm development. And if we look at the endosperm, what we can see with our indentation and extracting the pressure of the endosperm is that in the mutant, you have higher pressure compared to the wild type. In the wild type, pressure is decreasing over time. In the mutant, it remains quite high. But then the funny thing about this mutant is that the higher pressure doesn't make the seed bigger. On the contrary, you have more pressure inside, but you have a smaller seed. So we ask, can our model actually predict the same behavior, that increasing pressure will actually decrease growth, which is kind of unexpected actually. So what we did is that we took our simulation and we fitted our model with the very low drop of pressure that we see in the mutant, because initially our model was actually done at constant pressure, and we extracted the simulation that would best fit the data we had for the mutant. Okay? And then we took those simulations, we didn't change any parameters, and we just changed the pressure input in the simulation to pass from pressure of mutant to pressure of wild type. So we decrease pressure in simulation, and as you can see, as we decrease pressure, we increase growth, which is not at all a feature that we put initially in our model. So the model behaves like the experiments. You increase pressure, you decrease growth. So probably you have figured out why. It's because we don't have a simple balloon. Simple balloon would just have that top. We increase pressure, it's going to grow, basically. But then we have that function here, which is that the more pressure you have at the same time, the more tension you have in the seed coat, and that's going to stiffen the seed coat. And if you look at the stiffness of the seed coat in our simulation, as we are decreasing pressure from IQ2 to the Y type, we are delaying the stiffening of the seed coat, and that explains why you have more growth after, because you delay that inhibitory role here. And so we try to come back to the experiment and see, can we see that occurring in the mutant experimentally? So first, we looked if we had indeed more tension that would lead to a higher mechanical response of the cell of the seed coat, because we have a mechanosensitive gene whose expression can be induced if you compress the seed, for instance. So that's called LR1 that I showed you before, and you can see in the mutant, you have a higher expression of your mechanosensitive gene. So the cells are sensing that there is more tension, basically. And then if we look also at the changes in wall composition, so wall free again, so 1, 2, 3, 4, if we quantify, we see that we have a precocious accumulation of demethylesterified pectins in the cell wall of those mutants. So here, that's interesting, that works well. The problem we have in the plant field as well is the cell wall is a very complex material. So looking at the changes of composition is nice, but you can't really predict necessarily that is going to make your wall stiffer. And another problem we have here in the seed is that we are looking at an internal layer. So we can't use the IFM because the IFM is surface, basically. So we couldn't really measure the real mechanical properties, but we did a very simple experiment. We took our nanoadductor, we changed the tip to make a sharp tip, and we applied the same force, a given force, on the seed of the Y type and the mutant to see with a given force, is it easy or not to pierce my cell wall, wall 1, wall 2, wall 3, which we check by confocal microscopy, in the Y type and in the mutant at a stage where the mutant has more demethylesterified pectins. And as you can see, it's actually the wall free in the mutant is more resistant to rupture. So that doesn't really tell you that it's more extensible. That's not the extensibility. But at least it tells you that those changes in composition tend to change something in the mechanical properties of the seed. And so based on those data, it makes us kind of rethink the contribution of pressure. So because in textbook, it's just written, oh, pressure is a passive force for growth. It's all about the cell wall. Here you can see it's still all about the cell wall, but pressure needs to be tightly regulated during seed development. Because if you maintain a high pressure, you're going to have strong tension in the seed coat. The cells are going to sense it. They are going to stiffen early, and you're going to make small seeds. And in the Y type, as you reduce pressure over time, you maintain low level of tension so that the cell don't stiffen too early, and you make a bigger seeds. You can go further in that model, so that's from the mutant and the Y type, to look at that relationship between pressure and growth. So in simulation, it's very easy. In simulation, you can do whatever you want, obviously. You can look in simulation at the relation between pressure and growth and pressure and stiffness of a shell. As you can see, it's kind of the same thing we saw before. As we increase pressure, we are going to initially grow faster, but quite quickly we have an inhibition of growth and the seeds are smaller. And if you look at the stiffness, it's because you are going to have a precocious stiffening of the shell in the simulation. So experimentally, you can actually reproduce that, because what we do is we can actually cultivate Arabidopsis fruit in a tube. And if you increase the osmolarity of a medium by adding sorbitol in that medium, basically you expect that you are going to decrease the pressure inside the seed, because it's always, the pressure comes from a differential in osmolyte concentration between the outside and the inside. So our seeds are growing very happily in this medium, which is kind of surprising. And if we see the effect of sorbitol, we see that as we increase orbital concentration to decrease pressure inside the seeds are going to grow less fast. Initially, they are smaller, but they continue to grow much later than they would do actually in vivo. And at the end, they are bigger seeds. So you reproduce pretty much what you see in the model. Okay? So as you can see, that mechanical model that I can put back here allows us to predict, to explain how seed size is determined in Arabidopsis. And you would see there is that kind of model might actually work in any organism where you would have such a functioning like a shell, basically. Then we ask another question, is what about shape? So as I told you before, shape, you could find it boring for seeds. It's just a flattened egg, it's not much. But actually, if you look at the dynamics of seed shape changes in Arabidopsis, you see you have two phases. So if you measure the aspect ratio, so how much it's elongated, so we measure the vertical compared to horizontal there, okay? You can see that you have two phases. Initially, the cell is going to elongate along its main axis, so anisotropic growth. And then it's going to have a more isotropic growth. There is a slight inflation more in that direction compared to the other, but you have two phases of development, anisotropic growth and then isotropic growth. And then we ask, can that kind of model or mechanical interaction between seed coat and endosperm also explain that kind of behavior? So obviously, in this model, you see here pressure. The endosperm is contributing through cell growth through pressure. And by definition, pressure is isotropic. Okay? It's going to push in every direction the same. So if you look, then shape should depend on seed coat properties. You can check that in the seed very easily because, as I told you at the very beginning, the seeds are kind of a chimera, a natural chimera. The genome of the inside is different from the genome of the outside because the inside is product of fertilization, but not the outside. So if you take mutants of seed shape, so like those two mutants of microtubule-associated protein, you have catanine and spiral 2. So catanine is making round seeds, and spiral 2 is making elongated seeds. If you pollinate them with white-type pollen, the endosperm is going to be heterozygous, which usually doesn't give phenotype. But then the seed coat is going to stay mutant. And as you can see, the shape of the seed is the same as seeds that were self-pollinated. And that supports that it's the seed coat, which makes sense because it's cell wall properties, that is controlling the shape of the seed. Moreover, we have mutants. We have many mutants of seed size and seed shape. And we have a very nice mutant, which is called apetala2. That's a mutant for a gene, which is a transcription factor that is controlling the identity of the outer integument, so the two first layers of the seed coat. So if you look at late stage of development, they have differentiated. The first layer have those weird purple blob there, which are mucilage. So it's a kind of a gel that is inside that's going to be released when the seed is going to germinate. And then you see our thickening of our internal wall free there. And you see in that mutant, everything is gone there because those two layers did not differentiate. And as you can see, the seed is completely round. So except if you have some weird indirect effect of changing the outer integument, influencing the inner integument, it's kind of logical to predict that it's at least one of those two layers that is controlling seed shape. Okay? You may think, it's just about, all is about that wall free here. I told you that wall free is restricting growth. It's controlling the size. It should control the shape. But what's important is that that differentiation of wall free starts around 3 days. So it starts after the anisotropic growth phase. So before that phase, we don't know which layer of those two is controlling growth. So to look more at those two layers, first we look at the growth at cellular scale. And for that, so the PhD student who is working on all those seed shape aspects, Amelie Bauer, she developed a protocol so that we could extract the seeds from the fruit and cultivate them in vitro. You can see there, they are nice, they grow. They can even germinate to make another seed, another plant afterwards. And we can use confocal microscopy with a membrane marker and segmentation software called morphographics so that we can segment the surface of the first layer, the surface of the second layer of the cell. And if we have multiple time points, we can follow growth over time. And if we do that, we get map of growth. So here it's a map of growth that's projected on the 2 days after pollination, but the color represents how much each cell is growing from 1 to 2 days. They are also dividing, actually. And what you could see is that the growth is not homogeneous of a seed. It's actually the top half of a seed that is growing. So the two layers are doing the same, which makes sense because they're stuck together through the cell wall, so they can't be really disconnected. And what you can see here as well is that those cells here are elongating along this main axis because it's like the organ is doing. So what we need to explain is basically why are those cells here elongating along this main axis, which one and which layer is actually controlling that and determining the entire organ, the way it's growing. And so in plants, the control of cell direction depends on the orientation of the cellulose microfibrils because they are the load-bearing elements in the wall. If the cellulose are along a particular direction, like there, the cells grow perpendicularly because they prevent the growth along that direction. If they don't have any particular orientation, like there, they will grow isotropically. And the direction of cellulose deposition is controlled by elements of the cytoskeleton, which are the microtubules, so cortical microtubules. So there are microtubules that are just under the membrane. And what happens is that those microtubules are used as tracks for the displacement of transmembrane complex, which are called cellulose synthase complex, that are the ones that are synthesizing the cellulose fibers. So they just move along the microtubule, synthesize cellulose fibers, so that cellulose fibers are deposited according to microtubules. And so what you can do then is look at the organization of the microtubules in a plant organ to kind of predict in which direction the cellulose will be deposited. And you can quantify that using a tool called FibriTool that was developed by Areski Boudaoud. And so you see in the tool what you have, so in each shell you have a microtubules, you segment the cell with morphographics, and then in each shell the tool is going to calculate what's the mean orientation of microtubule fibers in the cell, and the longer the bar, the more it means that they're organized. You see in those cells it means most of microtubules are along that direction. In those cells the microtubules are more roundish, so the bar is small. So you have an idea of the orientation and the degree of organization of microtubules. So then for the seed what we did is then we use markers for microtubules, so microtubule reporters that would be only expressed in the first layer or in the second layer, so that we could look at the outer face or the inner face of the cell, which at the organization of microtubules, and how is it oriented in regards to the orientation of the seed. And what we could see is that in the first layer, microtubules in both faces tend to be more oriented perpendicularly to the main axis of the seed, whereas in the second layer, they are more randomly organized. So they should be the ones that are actually controlling the elongation perpendicularly to this axis through the guidance of cellulose synthase. So in this stage, you see that the first layer that is probably controlling the growth of the seed. So then, obviously, the question is, why are those microtubules organized like that? So, as I showed you at the very beginning, microtubules can organize according to mechanical forces in plants. They are even used often as a way to sense, to map mechanical forces in tissues. And for instance, if you take a developing meristem, so it's a bud that is producing organs, the shape of the bud is going to give to a specific pattern of forces. So like isotropic force here you have tension along this axis, here you have a tension along this axis. The microtubules orient according to that. And so we look whether actually our microtubule in the seed, in the first layer, would orient according to shape-driven stresses like they do in that bud. And to do that, you can see they correlate, the orientation correlate with the shape, so with the curvature of the seed. With that flat, so because again the seed is a flattened egg, what you have here is you have more curvature on the side there, which is the main axis of curvature is along this axis, even there. And if you correlate at cellular scale the microtubule organization and what's the main axis of curvature, you see that you have a very strong correlation between the two. You see that there is some noise though, and actually we didn't really care about that until very recently, so those data are actually not really treated yet, where Amélie has looked also at the microtubule dynamics. Because you know in animals, you know that microtubules are extremely dynamic. When we look at growth, we are looking at, in the plants here, at things that occur within a whole day. And actually what we see is that the microtubules are actually dynamic in the cell. You see here the same cell, over the course of 2 hours, it can completely reorganize those microtubules. Still, we have that correlation here. So we need to do a quantification though, but what actually I think happens is that those microtubules do orient according to shape-driven stress, but they are probably fluctuating along this orientation, and that we can measure. And probably if we integrate the behavior over time, we might have an even better correlation to shape-driven stresses. Okay? Okay, so the microtubules seem to orient according to shape-driven stresses. We can check that in our system they respond to mechanical forces. So what we can do, we can compress the seed, and we see that that increases the degree of organization. It doesn't work that well, probably because they're already organized. So it's hard to organize them even more. What you can also do, so again, that's not very helpful. That's a new data, so it's not processed yet. So that's a maximum projection, which is harder to see microtubules. But normally, if you do ablation, like we saw this morning, you're supposed to have a circumferential reorientation of microtubule around the ablation. And I hope it's not like just believing me, but there are microtubules that reorient there after 5 hours of ablation, even if it's hard to do because it's not projected properly. So that supports that microtubule can respond to forces. And then to show that that response of microtubule to forces through cellulose deposition is controlling seed growth anisotropy, we characterize the growth of many mutants. So here I'm just showing you the final shape of a mutant, but you see if you alter microtubule organization and response to forces, if you alter the guidance of cellulose synthase by microtubule or even cellulose deposition, you can completely change the shape of a seed. So that shows that the early anisotropic growth of a seed depends on microtubule response to forces in the first layer of the outer integument. Okay? So that beginning there is controlled by the first layer, okay, and by microtubule response to force. Then the question is obviously why do you have that transition from anisotropic to isotropic growth? Obviously, I put back that scheme here because you could say we know that that's the time at which the second layer is going to stiffen. If that wall free is going to be load-bearing instead of the first wall, it should be the one that is controlling also the shape. And that we made the hypothesis that that's our wall-free stiffening, which is also dependent on forces, that control the transition from anisotropic to isotropic growth. That actually would make sense if you look at data. So if you look here at around 5 days when the walls have started to differentiate and that wall free is becoming load-bearing, if we look at the microtubule organization in the face of a cell that's facing that wall, you can see that microtubules are very, very disorganized. They have a tendency to be aligned actually a bit in parallel to the main axis of the seed, but anyway, they don't have a very strong organization. So they wouldn't give a big bias in anisotropy in mechanical properties. But what you would say is that if they guide cellulose synthesis, that shouldn't be in any particular direction. That fits with the idea that you have isotropic growth. Yep, okay, I'm done actually almost. But then there is a problem actually, because if we go back to the data on growth, so on seed size and shape, if you derive seed size and seed shape to get the growth anisotropy and growth rate, you see that growth anisotropy decreases very strongly from the beginning, and it's almost null at 3 days, whereas seed growth rate decreases much later. If both processes would be controlled by that differentiation, you would not expect that to occur before that. You would expect them to occur at the same time. And so we looked then back at the first layer, and what we could see is that actually what we see in the first layer is that the microtubule are getting disorganized also over time. Here we have actually an early time and a late time, but if we look at every day, now we can, the microtubule in the first layer, they very quickly disorganize. And they're disorganized at the same pace you see the growth anisotropy decreasing. So the inhibition of anisotropy is actually probably due to a reduced organization of microtubule according to shape-driven stress. And that makes isotropic growth. You can see in this map of growth, the growth is completely isotropic. The cells look like soap bubble, basically. But the funny thing there that we can't really understand is that the microtubule get disorganized, but it's not because they can't respond to forces anymore, because if we compress the seed, if we apply mechanical forces, they very strongly respond to that application of mechanical forces. So they haven't loosened their ability to respond to forces, but still they don't align according to shape-driven stresses anymore. And so from that, I can conclude with a model showing you that basically I've shown you that both the size and the shape of a seed depend on two mechanical responses that are occurring in two different layers of a seed coat. You have a pressure that's going to lead to some tension, that's going to organize microtubule according to shape-driven stresses in the first layer. That's going to promote growth anisotropy. But then that response is actually dampened over time. We don't really know why. But then at later stages, it's going to stiffen the wall free, so because of the mechanical response of the second layer, and that's going to inhibit growth rate and also make growth also isotropic at later stages. And just to conclude, just to show you where we are heading now, so now with Olivier Ali, we are trying now to implement those two mechanical response in model, go back to the modeling, to do spatial models on the entire seed. So that's an ovule actually, so at 0 day, but we can do it at later stages as well, segment fully the entire organ in 3D and then do simulation. So that's just a shell simulation with the outermost cell wall, but the idea is to do multi-layer simulation. And then one thing that's very important that we're missing now is to have real measurement of mechanical properties. And now I'm starting to collaborate with a team of Torsten Harman in Norway, and they are using BRI1 microscopy to get a way of having a way to have viscoelastic properties of internal cell wall. And then finally, you can also use a FLIM probe that has been developed to have an idea of the microviscosity of the different cell compartments in developing tissues. And I think that's really promising because that should help us to have more biophysical measurements in living tissue over time, basically. And with that, I will finish by thanking all the people of the RDP laboratory, and then especially Audrey Kreff, who did most of the experimental work on the seed size, Amélie Bauer and Camille Bier, who do the work on the seed shape and size as well, and then Olivier Ali for developing the mechanical model.
Yeah, thank you for this really nice talk. I had two questions, so I may try to go fast. The first one is, do you have any idea of the origin of the turgor pressure and the maintenance of turgor pressure and growth? Yes. And maybe the second one is, in the model by Olivier, do you have changes in the mechanical properties of the because of the age and not because of the size, for example? Yeah. Do you see?
I see exactly. Changes? So for the first one, turgor pressure is osmolyte accumulation in the seed. That's sugar osmolyte mostly. We've measured that in IQ2 as well and we have changes in ratio between glucose, fructose, sucrose, basically. And I find that super interesting because then that relates control of mechanical properties, so turgor pressure, with metabolism. Because the seed wants to accumulate sugar, it's going to be converted in oil afterwards, but that is going to influence its mechanical properties. And then for the second one, I completely agree with you, and I think that's what happened, why those microtubules lose their organization according to shape-driven stresses. I think that the response of microtubule to forces is at the cell wall interface. We don't really know how it works at the moment in terms of sensing, but I think that if you change our mechanical properties of our cell wall, because of our composition, that's going to change the response. And that's probably why our response is dampened over time. We know that those cell walls we have done immuno have changes in composition at the same time as the microtubule gets disorganized, basically.